Hello everyone, let's get started. Today we're going to begin the first of a two-part lecture series about the brain. Together with the spinal cord, the brain is part of the central nervous system. The brain functions as the primary control center for the nervous system. It receives and integrates all of the incoming sensory information from the peripheral nervous system, and it determines the appropriate response, which it sends out through motor pathways along to effector cells. Like the spinal cord, the brain contains two cell types, both neurons and glial cells. In terms of gross anatomy, the brain and the spinal cord contain gray matter and white matter. The first thing to know about the gross anatomy of the brain is that the gray matter and white matter of the brain are organized differently than what we saw in the spinal cord. Remember, in the spinal cord, the gray matter was in the center and the white matter surrounded it. We're going to see something different in the brain. The gray matter is still a collection of neuron cell bodies and the dendrites that extend off the cell membrane. And like the spinal cord, the reason that gray matter is gray in the brain is because of pigment, both melanin and lipofuscin. We talk about gray matter as cortices in the brain. That's the plural form of that word. The singular is cortex. So we talk about the cerebral cortex in the brain. That is the gray matter layer that lies on top of the white matter. Now, gray matter can also be found deep within the white matter in regions that we call nuclei. These are deeper masses, and they are found in very specific locations inside of the white matter of the brain. This term cortex is translated as bark, and one of the ways to remember that gray matter surrounds white matter in the brain is by remembering that bark surrounds the circumference of the tree. In terms of white matter in the brain, just as we saw in the spinal cord, the white matter is composed of a collection of axons, and it is distributed in structural regions inside of the brain. It's also organized functionally in tracts, just as we saw in the spinal cord. Remember, while we talk a lot about the neurons that are inside of the brain, there are also an abundance of glial cells as well. On this slide, up at the top, you're looking at a drawing of the human brain. You can see that they have taken a section through the brain, and then we're shown that face, that cross section of the tissue. They're outlining for us, at least at this point in the brain, the distribution of the gray matter in the cortices and the white matter underneath. Remember, in the spinal cord, the organization is opposite to that. The gray matter is deep within the cord, and the white matter surrounds it. The white matter that lies in the brain has different myelination patterns than the white matter that we see out in the peripheral nervous system, what we would call nerves. We've talked about myelination in terms of what myelin is made out of and what cell types secrete it. We've talked about why the certain axons are wrapped in myelin, what function that myelin provides. But we haven't yet talked about how myelination differs from the central nervous system compared to the peripheral nervous system. Remember, myelin is a fatty material. It provides insulation around bundles of axons in the nervous system. It helps to increase the speed of transmission along these neural pathways without loss of signal. We've talked about how the diameter of the nerve out in the peripheral nervous system is critical. The wider the nerve, the more membrane surface area it has. And then that membrane surface area provides for faster transmission of these nerve impulses. We said in the peripheral nervous system, myelin is being secreted by Schwann cells that literally wrap themselves around individual axons. We said that in the central nervous system, myelination is being performed by oligodendrocytes, similar type of cell, but rather than wrapping itself around an axon, it sits nearby and sends out membrane extensions to secrete the myelin. Now let's take a look at some of the differences in myelination between those two places in the body. Out in the peripheral nervous system, the diameter of axons ranges from about 0.1 to about 20 micrometers. In comparison, in the central nervous system, axon diameter ranges from about 0.1 to only 9 micrometers. So the diameter of axons inside of the central nervous system is significantly smaller than what we see out in the periphery. In the peripheral system, any axons that are less than about 2 micrometers are left unmyelinated. The diameter of those axons is small enough that they are left unmyelinated. Axons that are greater than 2 micrometers in the periphery will be myelinated by those Schwann cells. Now look at the numbers in the central nervous system. Again, axons less than a certain size are going to be left unmyelinated, but look at that size. Anything less than 0.2 micrometers is going to be left unmyelinated. Anything greater than that is going to be myelinated. So in the central nervous system, many more axons are going to be surrounded with myelin than what we find in the peripheral nervous system. And if you think about it, this makes sense. Speed of transmission in the central nervous system could be thought of as being more critical. We have a lot more information coming in. That information has to be integrated. It has to be not only collected from a variety of cells, but it has to be passed between a variety of cells before it's then sent back out. Speed of transmission is critical there. The other thing to remember about myelination is that when axons travel down an unmyelinated axon, the action potential has to be regenerated along the length of the axon, but there is no need for saltatory propagation because there is no myelin. We do see axon regeneration down the length of the axon, so we do see propagation, but we can describe that propagation as occurring sort of like a wave. We see a wave of action potential regeneration occurring down the length of the axon, one after the other after the other. Compare that to what we see with a myelinated axon, where saltatory propagation occurs. And remember, that means that the action potential has to travel under the length of the myelin region, and then the action potential gets regenerated at the node, travels down underneath the myelin, gets regenerated at the next node, We describe this as saltatory, or you can think of it as hopping from one node to the next, to the next, to the next. So more like a wave pattern in an unmyelinated axon, more like a hopping pattern in a myelinated axon. So we can easily identify the regions of gray matter and white matter in the brain. We can also easily identify very grossly visible regions of the brain. The cerebrum, for example. This is the largest region of the brain. There's also a region towards the back of the brain that we call the cerebellum. And then if we take the cerebrum away, underneath the cerebrum, tucked sort of underneath, is what we refer to as the brain stem. The cerebrum is the largest division of the brain, size-wise. Second comes the cerebellum, and then third comes the brain stem. Now, it's important to remember that there are other structures in the brain, not just cerebrum, cerebellum, brainstem. And we're going to be talking about those structures and what they do. So we said the cerebrum is the largest physical structure. It makes up about 83% of the volume of the brain. It's composed of two hemispheres. And those two hemispheres are connected to each other with a structure that's called the corpus callosum. As I'm sure you know, the surface of the cerebrum is not smooth and flat, it's convoluted. It's covered in these folds of tissue that are separated from each other by shallow grooves. Now, each one of these folds of tissue is called a gyrus. The plural of that term is gyri. And each one of those shallow grooves is referred to as a sulcus. The plural of that term is sulci. So the entire surface of the cerebrum is covered in gyri and sulci, folds and grooves. The cerebellum is about 10% of the volume of the brain, so a much smaller part of the brain. However, it contains about half of the neurons in the brain. The brainstem makes up the remaining volume of the brain, about 7%, and the brainstem is composed of three separate structures: the midbrain, the pons, and the medulla oblongata. The brain stem actually ends right at the foramen magnum in the skull, and distal to that is spinal cord. On this slide, we're looking at both a fresh specimen of a human brain and a preserved specimen. The fresh specimen is laid out on a flat surface, so it doesn't quite have its normal anatomy, but you can see the two hemispheres laid out next to each other. You can see the cerebellum that has sort of flopped over to the side here, and you can see a portion of the spinal cord. Can't really see much of the brainstem. It's lying underneath the two larger cerebral hemispheres. A fresh brain specimen is very fragile to handle. If you were to try to pick it up, you'd have to be very careful because it's friable. It can fall apart in your hands. The tissue is very soft and it's very fatty. It's very oily or buttery feeling in your hands. When you take a fresh brain out of a skull, it sort of flops onto whatever surface you lay it onto. It doesn't stay in the same anatomical position that it has in the skull. You can see that positioning over here in the drawing on the upper left. Now we're looking at one side here. We're looking at a lateral surface of one side, but you can see how the cerebrum normally sits in relation to the cerebellum, which is located inferior and posterior, and the brainstem, which is also on the inferior surface of the brain, more centrally located, aligned with the spinal cord. You can see the same structures in the preserved brain on the lower left. Again, we're looking at a lateral view here. We can see the cerebellum here. Can't see much of the brainstem, but it would be located right around here, and it continues down into the spinal cord. One of the most remarkable features of the brain, whether you're looking at a fresh specimen like this one or a preserved specimen like this one, is the size of the two hemispheres in relation to the other parts of the brain, and also this pattern of gyri and sulci. One of the questions that I get from students a lot is why? Why does the brain have this bizarre infolded pattern of gyri and sulci? The answer to that question is surface area. Surface area. You're trying to fit an awful lot of cells into a limited space inside the human skull. Remember, the gray matter, which is in the area of the cerebral cortex, the bark, the outside layer, the gray matter is composed of neuron cell bodies. And in order to pack as many cell bodies in as possible into that cortical region, the only way to do it is to fold that tissue up. You get an increased surface area that way, more cells in the same size space. If it had a flat, smooth surface, you just wouldn't be able to fit as many cells in. So it's all about surface area. When we talked about the spinal cord, we talked about the meningeal layers that surround it. And we said that those same meningeal layers continue upwards to surround the brain as well. Remember, there are 3 layers to these coverings. There's the dura mater, the outermost layer, the arachnoid mater, that's the one in the middle, and the pia mater, that's the one that lies innermost or closest to the brain. There are a few differences to make note of in those layers as they relate to the brain, and that's what we'll go over next. The dura mater, the outermost layer, can actually be described as having 2 sublayers, a periosteal layer and a meningeal layer. The periosteal layer of the dura mater is the side that faces the skull. It's interesting because the dura mater is actually attached to the bony skull, but only in a few spots. It tends to be attached along the suture lines of the skull and also down in the area of the foramen magnum, but it's not attached all over. Now, the inner layer of the dura mater is the meningeal layer. That's the side that faces the arachnoid mater. The 2 layers of the dura mater just lay right on top of each other, but there are some areas where they don't lay on top of each other. There are some areas that they are allowed to be separate from each other, and those regions are referred to as dural venous sinuses. I often tell students to imagine you have 2 pieces of cloth laying on top of each other, and you run 2 lines of stitches, maybe an inch apart from each other. What you've created between those 2 lines of stitches is like a little channel You could put your finger in there, or you could put a pencil in there. That channel is very similar to these dural venous sinuses. Now, these sinuses have the job of draining venous blood from the cranium, from the head, into the jugular vein. They are not veins. They are venous sinuses. They're not made out of the same tissue that veins are made out of. They are made out of two of the layers that make up the dura mater. That's what they're made out of. But they have the job of serving as passageways or channels that direct venous blood back to the jugular vein. Now, in between the meningeal layer of the dura mater and the next layer of the meninges, which is the arachnoid mater, in between those two layers, there is a bit of space. Now, the meningeal layer of the dura mater and the arachnoid mater are attached to each other in a couple of places, but otherwise, we have laid those two layers on top of each other, and there's a little bit of space in between them, enough space for cerebrospinal fluid to flow between those two layers. We give this space a name. We call it the subdural space. You may have heard of the term subdural hematoma. That is a form of brain injury where blood starts to pool in the subdural space, forming what we call a hematoma. That is a medical emergency because there shouldn't be blood in the subdural space. There should only be a little bit of cerebrospinal fluid. Now, underneath the dura mater is the arachnoid mater. This layer of tissue looks a lot like it does as it lays over the spinal cord. It's composed of a layer of simple squamous epithelium. It faces the meningeal layer of the dura mater on one side, and on the other side, it has that meshwork of fibers extending off of it and reaching towards the pia mater. Remember, that is subarachnoid space, and that is another space that is full of cerebrospinal fluid. Finally, we have the pia mater, the innermost layer. This also looks a lot like it looks around the spinal cord. It's very delicate, it's vascular, and it closely follows the contours of the surface of the brain. On this slide, you're looking at a drawing of a human brain, a lateral view. You can see the cerebrum in pink. Here's the cerebellum. Here's the brainstem. And they've cut it off right where it would become spinal cord. What you're looking at here are these venous sinuses. Look at how they travel across the surface, the outer surface of the brain. They've also drawn in for us where the internal jugular veins would be. Remember, these venous sinuses drain into the jugular veins. On this slide, we're looking at a small section through the skull and through the meninges. So here's a lateral view of a human head. You can see this little box drawn right here. They've taken that piece and they've blown it up for us right here. What you're looking at right here, this is brain. You can see the cerebral cortex, the gray matter, overlaying the white matter. This would be a gyrus. This would be a sulci. Another gyrus, another fold, another sulci. If we look over to the other side, Of course, the outermost layer of tissue would be skin. And underneath that skin, we would find bone, would find the bone of the skull. You can see they've drawn in the periosteum that would cover that bone. There would be, on the other side, the 3 layers of the meninges. So what they've drawn in here, and I know it's a little hard to see, but they've drawn in some gray on top of some blue. The gray represents the periosteal layer of the dura mater. So that's the part of the dura mater that lies right against the bone of the skull. The meningeal layer of the dura mater sits on top of the arachnoid mater. So this line right here, this whitish gray line, would be arachnoid mater, single layer of simple squamous epithelium. One side is next to the meningeal layer. One side has these mesh fibers coming off of it. That is the subarachnoid space. And after that space comes the pia mater, the layer of tissue, the vascular, delicate layer of tissue that lies right on top of the brain. If you look on the lower part of this slide, you can see a cross section through the human brain. You can see the cerebral cortices in the darker color. That's the gray matter. You can see the white matter underneath it. Now this brain has some very strange looking anatomy. We don't see that nice mirror symmetry that we normally see. We can't clearly see the 2 hemispheres here, and that's because of this. This right here represents a subdural hematoma. So this is a collection of blood in between the meningeal layer of the dura mater, that's the bright blue, and the pia mater. Remember, normally there's a little bit of cerebrospinal fluid in there, but not this volume of blood. See what this does to the brain tissue. It distorts it. It pushes against it. And the brain is a soft, squishy tissue. So it has nowhere to go. It's surrounded by skull. And what ends up happening is this tissue, this brain tissue, gets pushed, and it cannot function normally if it is squished into a smaller area. And that person would show signs, neurologic symptoms and signs that their brain was not functioning normally. Speaking of blood, let's take a minute now and talk about the blood supply to the brain. It actually comes from 4 large arteries. There are left and right common carotid arteries that feed the brain. And then there are left and right vertebral arteries that feed the brain. Remember, arteries are carrying oxygenated blood. So those 4 arteries are carrying oxygenated blood toward the brain. They're then going to branch off into smaller and smaller and smaller vessels. Arteries become arterioles. Arterioles become capillaries. We see the exchange of gases at the capillary level between the blood and the nervous tissue. Once the oxygenated blood becomes deoxygenated, now it's moving on the other side of the circulatory system. Those capillaries that are now carrying deoxygenated blood will become venules, and those venules will get larger into veins, and that blood will be taken back towards the lungs for reoxygenation. Remember, it's the pia mater that is the vascular part of the meninges. It's the pia mater that contains all of those branching blood vessels that supply the brain. The brain is very metabolically active. Of course it is. Your brain works 24 hours a day, 7 days a week, 365 days a year throughout your entire life. And it does a lot of integration work. So it's very metabolically active. 15% of the entire blood supply in your body is feeding the brain at any one moment. It takes up about 20% of the oxygen and the glucose in the body. It has a lot of needs for energy. You can see this when you see situations where blood supply to the brain is interrupted. You can see what happens when it can't get those energy needs met, when it doesn't get the oxygen it needs, when it doesn't get the glucose it needs. If you were to, for example, cut off the blood supply in 2 of these arteries. Let's say, for example, someone was choking a person and cutting off the flow of the carotid arteries on both the left and right side of the neck. Within a few seconds, that person would lose consciousness. Within a couple of minutes, we would start to see impairment in the brain. We would start to see that the functions that the brain performs, receiving, integrating, and then eliciting a response on the motor side, it would not be doing those functions normally. Those would be impaired. And of course, after about 4 minutes or so, depending upon how old the person is and how healthy they are and so on, after about 4 minutes or so, we would begin to see irreversible death of the cells in the brain, both the neurons and the glial cells.
You could argue that the brain is the single most important organ in the human body. So it would make sense that the body takes special steps to protect this vital organ from things that might otherwise harm it. And that brings us to a discussion of the barriers that are in place around the brain to protect it. The blood-brain barrier, or the BBB, is the barrier that helps prevent substances that might be circulating in the blood from gaining entry to the neurons if they could harm those neurons in any way. Remember that blood feeds the brain. It comes up through the blood vessels that we've discussed, enters into smaller and smaller vessels until finally it goes into the capillaries that are most closely associated with brain tissue. Substances can exit those capillaries by crossing through the membrane of the endothelial cells, the cells that line blood vessels. And then those substances enter into the space outside the capillaries, the extracellular space. From there, those substances could cross into neurons, but they're prevented from doing that by a specialized kind of glial cell called the astrocyte. From that extracellular space, any substance that leaves the bloodstream is going to cross into the astrocyte first before it can cross into a neuron. The astrocyte membrane, it turns out, has a very low permeability. Remember what permeability is. It's a measure of how easy it is for something to come across. So a membrane that has a low permeability doesn't allow very much to come across. The astrocyte membrane is fairly permeable to things like water and glucose and lipid-soluble things like oxygen gas and carbon dioxide gas, even things like alcohol, caffeine, nicotine, various anesthetics. Those things can can come across the astrocyte membrane and then gain entrance into the brain tissue. But other things are prevented from going into the neurons because of those astrocytes. They create this barrier to entry around the brain, helping to protect the brain from things that might be toxic to it. Now, there is one other barrier we need to be familiar with, and it's the blood CSF barrier, the barrier between the bloodstream and the cerebrospinal fluid that the brain is bathed in. Remember the choroid plexuses. This is another possible point of entry into the brain. Remember that those choroid plexuses are inside the ventricles. They're laying on the lining of the ventricles. But between the choroid plexus and the cerebrospinal fluid, there's a layer of cells. Those are the ependymal cells. Remember, they are a specialized type of epithelium. And like all epithelial tissue, the cells are closely packed together and connected to each other with tight junctions. So it would be very difficult for a substance to get past this epithelium unless it can cross over the membrane. Ependymal cells help produce CSF, remember. The blood gets filtered through these ependymal cells to help create the cerebrospinal fluid. And in that process, the ependymal cells are going to prevent certain substances from getting into the CSF. They form another very important barrier